Hello! For those of you who may not know me, my name is Laura Ritchie, and I earned my PhD in chemistry from Penn State, where I specifically studied RNA. Until this year, most people have not heard of RNA or know what it is. And so the nerdy scientific part of me is excited that people are starting to learn a little bit about it. With the Moderna and Pfizer RNA vaccines currently being discussed in the news, I wanted to give a little bit of background into what RNA is, the science behind RNA, and the science behind these vaccines. Please know that I am not getting paid to do this. I truly just want to inform people what is going on and how this works scientifically to perhaps help them make well-informed decisions. First, what is RNA in general? Well, the basic central dogma of molecular biology, or how our body accomplishes all of its tasks, starts with DNA that gets transcribed to RNA, which is then translated into proteins. DNA and proteins are more commonly known, whereas RNA is sometimes forgotten. When someone mentions the genome, it's the DNA that they are referring to. The DNA, as a simplification, lasts the length of the cell. It's very stable and the sequence never changes. Pieces of this sequence then get copied into RNA. It's almost like you would be transcribing someone else's notes into your own handwriting. This RNA then sends a message to the cell to tell it what to do, and then gets degraded. RNA actually degrades very easily and is one of the reasons it can be difficult, but not impossible to work with. The message that the RNA encodes is what is used to make the proteins. Proteins are then the functional tool. Let's use an analogy of something you may want to fix or figure out in your car. You could think of the DNA like the instruction manual to your car. You don't throw it away, but if there's just one part of the car that you're interested in, you don't look at most of the instruction manual. It's also a lot easier to find a short YouTube video that helps you do exactly what you need to do with your car. That's what the RNA is, this short instruction video. Then the protein would be the part of the car that you're using. Very importantly, in normal human biology, there is no way to go backwards from proteins to RNA or RNA to DNA. Other organisms and certain viral infections can cause a backward process, but in our normal cells, that is just not the case. Before discussing the vaccine, we need to be on the same page concerning the SARS-CoV-2 virus. This virus is an RNA virus. This means that there is no DNA in the virus, it is just RNA. The RNA enters our cells, and because it has features that look like our native RNA, it uses our cells to make proteins. I'm circling some of them here. In fact, there are about 29 different proteins that get made from the single piece of RNA. I could spend a lot of time talking about how this works because I find the RNA chemistry of this fascinating, but it is beyond the scope of this video. And these different proteins and then another piece of RNA then get packaged into a viral particle and then can infect other cells. One of these proteins is the spike protein, sometimes abbreviated the S protein. When an individual is able to fight off the SARS-CoV-2 virus, the antibodies produced are targeting this spike protein. This leads us to the RNA vaccine. This vaccine contains a piece of RNA that is the sequence that makes the spike protein. The spike protein gets made by our cells the same way that 1, we make normal proteins, and 2, the virus forces our cells to make the spike protein. This protein is then transferred to the surface of our cells, and our immune system makes antibodies to it. There's a lot more to the immune response that I'm not getting into, partially because I'm not an immunology expert. If you want to know more about this, I can refer you to people who are. Because RNA gets degraded in our cells so easily, once it causes the production of this protein, it will get degraded and will not last forever. Importantly, this will not cause COVID-19, as this RNA is not producing the whole virus, or is not even the whole viral RNA sequence. It's just the sequence that codes the single spike protein, none of those other 29 proteins. Also, this will not alter or change your genome. Because RNA is similar to DNA, some may think that RNA has the potential to change your genome. But this is RNA and your genome is DNA. Although they look similar and even sound similar, they behave extremely differently in your body. And like I said, our bodies have no way to convert RNA into DNA. My biggest concern with the vaccine development is not necessarily the science, because I know that the science is very sound. My biggest concern was the speed of development. Since typical vaccines take much longer, up to 10 years, to develop. However, typical vaccines are either weakened forms of the virus or a form of an important protein of the virus, like the spike protein. These both take much longer to develop. Weakened viruses take a while to develop because they must be weak enough to not cause the disease, but similar enough to convince your immune system to make the appropriate antibodies. Proteins are also more complicated to develop to be sure that the very complex three-dimensional structure is maintained. RNA, on the other hand, is much simpler to develop, as it is just the sequence that needs tweaked. So only the sequence for the spike protein needs to be added here, and then your cells make that spike protein. Essentially, the RNA vaccine is instructing your cells to make what would have been a typical vaccine. The sequence that coded for the spike protein was determined early on in the pandemic. Practically, as I was researching the speed of development, I was reminded that most of the 10-year vaccine development is actually spent securing funding rather than performing the testing, and this hurdle was cleared due to urgency. And then in terms of RNA treatments in general and why this is such a new thing, the biggest hurdle to deliver any RNA treatment is the delivery to the cells, not necessarily the design of the RNA originally. Because RNA is negatively charged, and our cells do not let anything in or out that are charged. Very recently, this hurdle had started to be overcome because there were huge advances in developing delivery systems for RNA treatments. In fact, Moderna even has another RNA vaccine for Zika in clinical trials. So all that was needed to be done to make this applicable to SARS-CoV-2 was to change the RNA sequence. The delivery mechanism, in this case a lipid nanoparticle, was already developed. Additionally, one of my favorite RNA treatments to look at the story behind is Spinraza. If you're interested, I would encourage you to look that up. So I hope that what I showed you helped you understand the science behind these new vaccines. To be honest, I was trying to find scientifically sound reasons to question the vaccine, but I wasn't able to, at least with the results that have been published so far. I am familiar with the work of some of the scientists at Moderna, and my advisor at Penn State is friends with some of them. They are great scientists and people, and I do trust their work. However, I know that this can be a complex topic, so please contact me if you have any questions. If we are friends on social media, reach out to me there, or email me at ler73@pitt.edu. I hope you enjoyed this video and that it answered some of your questions.